Hello, this is just a video to show how to design primers for tagging a gene in Japonicus using OpenCloning. So the way we would do this in yeast is we would use a plasmid, we would amplify some cassette from it using PCR and integrate it in the genome doing homologous recombination. So let's start by retrieving the starting sequences. Let's start, for example, by getting the plasmid. So we can get it from @gene. So in this case, I select a repository, @gene, and I put the identifier of the plasmid. In this case, this plasmid has a GFP and a kanamycin resistance cassette. I can see it also in the editor by clicking in the eye. And if I see this GFP, we see that the sequence it starts in here. Then let's load the genome sequence. So in this case, we could use a particular assembly that is not the reference. So here you provide the assembly identifier from the NCBI and you can load from any genome. But in this case, let's just do from the reference genome of Japonicus. And here we can query by gene name. So I'm going to look for the gene ACE1, which is my favorite gene, and I find it here. And I can request upstream and downstream bases for that, and I will get the sequence of the genome. And I know that Japonicus genome annotation is a bit wacky, so I guess this was annotated to be a whole gene, but actually it has 3 coding sequences inside. Anyway, doesn't really matter. We see that if we come here, we can click on this and we see that this coding sequence is actually the ACE1. If we double click, we see the annotation. So we know it's this one. And let's say now we want to design primers to tag this with GFP. So then we would come here, we would say we want to do a PCR. The purpose, we want to design primers, and the purpose of those primers are homologous recombination, and our target sequence is this one here. When you click on design primers, we will be asked what we want to amplify from the plasmid. So in this case, I'm going to select the GFP and the kanamycin resistance cassette. You can, move it until you want in this editor. I don't know exactly until when you want to amplify, but you can select it in here. And then you choose that, and then you'll be asked to select what you want to replace. So for example, if we wanted to do a deletion of this gene, we select the coding sequence and we choose that as the replace region. In this case, where we want to add a GFP tag, we can go to the sequence, to the end of the ATG, sorry, to the end of this coding sequence of ACE1, and we can either replace the codon, the stop codon by what we are going to amplify from the plasmid, or we could just select a position and it will do an insertion. Let's say we want to replace the stop codon. So you select the stop codon and then you click on choose region. So we have selected what we want to amplify from the plasmid, what we want to replace in the genome, and here we can provide other settings such as what is the homology length that I want for the primer, the TM that I want for the primer, and the minimum length that the primer should have for the hybridization. So let's just put 14 as a minimum. And then you can select whether the orientation of the insert is forward or reverse. In this case, we obviously want forward. And if you wanted to add some spacer between the insert and the genome, you could put them here. So if we go to the editor in this, when we are in other settings, we can see the sequence of the final product. So we can see it here. We see that we have the Japonicus gene and then we have the GFP. So we can verify that it's going to be inline by going to the sequence map. And we see that indeed we have our gene of Japonicus that then is in line with the start of the GFP. And we can just double check that the GFP, probably probably as an stop codon here, right? Yeah, that's it. Okay, so it's all good. Then we click on design primers, and these primers will be made. You can give them another name. By default, they're called forward and reverse. And when you click on save, they will be put on the PCR. And this will give you the sequence of the PCR product. You can see some details here, such as, you know, TMs, whether it forms heterodimers or not. If you want to see more details of the primer, you can also come here, and you will see some extra information, whether it forms a hairpin. All this comes from primer 3. And finally, you can do the homologous recombination and get the final product. The only issue here is that you won't keep the coding sequence annotation from this. You know, unfortunately, because you cut through the ATG here, it no longer shows you the coding sequence of the gene from the ATG to there. So you would have to annotate that by hand. Yeah, that's unfortunately not the case yet, but it will be supported in the future. So yeah, that's how you do it. And yeah, if you have some feedback or some comments, please let me know.